Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover Laboratory 27, the discussion portion. So, before we get into really what we did in the laboratory, we need to cover some background information. We need to cover some anatomy. Remember, form and function go hand in hand, and in the kidneys, it's no different. Form and function are very important. So let's talk big picture. An analogy that I've heard before about how the kidneys work, it's like cleaning your refrigerator. When you clean your refrigerator, you take everything out. You take your old mayonnaise, all the stuff that's been in your fridge for way too long. You pull everything out. You clean your refrigerator, and then you put the good stuff back into your refrigerator. Okay, you put the good stuff back in, you leave the old mayo out. Your kidneys work in a similar fashion. So let's look at some anatomy first. So we've got this capsule right here called Bowman's capsule. Within Bowman's capsule, we have a capillary bed, and this is a special capillary bed. It's called the glomerular capillaries or the glomerulus. So we have the afferent arteriole bringing blood to the glomerulus. The glomerulus sits inside Bowman's capsule. The blood flows out of this capillary bed into what's called the efferent arteriole. The efferent arteriole. And then it goes into another capillary bed, but this is another special capillary bed. It's called the peritubular capillaries. Why is it called the peritubular capillaries? Well, because it's around the tubules. This kind of looks like a tube, right? So this is a unique part of our circulatory system because it's a portal system. A portal system is when you have two capillary beds within the same kind of pathway of your circulatory system. So we have our first capillary bed, which is the glomerulus. We have our second capillary bed, which is all around here, which is the peritubular capillary. All right, let's go back to this Bowman's capsule. So this Bowman's capsule is right here. It leads into our proximal convoluted tube, which leads into our loop of Henle. Now there's two parts of our loop of Henle, the descending and ascending, descending and ascending. And then we have our distal convoluted tube, and then we have our collecting duct. All right, tubular elements. So these are our vascular elements: afferent arteriole, glomerulus, which is a capillary bed, efferent arteriole, peritubular capillaries, which are our second capillary bed, and then it goes to our renal vein. Tubule elements: Bowman's capsule, proximal convoluted tube. You'll also see this called the PCT. Then our loop of Henle, LOH, loop of Henle. We have our descending limb and our ascending limb. And then we have our distal convoluted tube. Sorry, this should say distal down here. Let me get my annotator, my pointer going, my spotlight. All right, down here, this should say distal. DCT, distal convoluted tube, and then our collecting duct. So, how is this form going to function to do all the things that kidneys need to do? Well, there's three main processes that are going to happen. And what ends up as urine is the result of these three processes. So we're going to break each one of these processes down. We're then going to put all of these processes together, and that's what's going to determine what ends up in the collecting duct, what ends up as urine, what gets excreted from our bodies. So, filtration. When we talk about the kidneys and we say filtration, we're going to talk about fluid leaving Bowman's capsule. So leaving the glomerulus and entering Bowman's capsule. So filtration is blood to the tubule lumen. Remember, lumen is like the inside of a chamber in the inside of a tube. So plasma is going to leak out of the glomerulus into the Bowman's capsule. Now, the glomerulus, it's selective. It's only going to let some things out. So most liquid components are going to leave the glomerulus. Ions are going to leave. Urea is going to leave. Water is going to leave. Some important things that will not leave at the point of the glomerulus, that will not be filtered out into Bowman's capsule under normal circumstances, are going to be proteins. Proteins, red blood cells do not leave, proteins do not leave, white blood cells do not leave the glomerulus. Glucose does leave the glomerulus. It does get filtered out into Bowman's capsule. Bowman's capsule is also known as the renal corpuscle. So things moving from the glomerulus into the nephron. That is filtration. Filtration is blood to tubule lumen. Filtration is glomerulus, this capillary bed, to the nephron, the Bowman's capsule part of the renal tubule system. That's filtration. Filtration is leaving the blood system. That's one process that we need to consider when thinking about what ends up becoming urine. If you leave the glomerulus and you enter the renal tubule system, then you're on the path to the collecting duct. And once something's in the collecting duct, it's urine. Reabsorption. Reabsorption is going to happen throughout the tubule system. And reabsorption refers to the movement of things from the tubule lumen to the blood. So, our fridge analogy. Up here at filtration, we take a ton of stuff out. We take basically everything out of our fridge. That's filtration. Reabsorption, we start putting good stuff back in. So glucose, for instance. Glucose is a valuable resource. We don't want to pee out all of the glucose that we have in our bodies. Glucose gets filtered out, but then it gets reabsorbed. Most reabsorption is going to happen in the proximal convoluted tube. So tubule lumen to blood, renal tubule, proximal convoluted tube. And it goes into the peritubule capillaries. And now let's look at secretion, the final process that's going to determine what eventually becomes urine. So secretion is going to happen primarily in distal convoluted tube. And our blood is going to be able to pass additional things that did not get filtered into the tubule lumen. So like penicillin is an example of something that gets secreted. So peritubular capillaries into the nephron. Peritubular capillaries into the nephron. Secretion. Those three are going to combine to determine what eventually becomes urine. All right, so the majority of filtered fluid is reabsorbed. So about 20% of the liquid that goes into the glomerulus is going to be filtered. It's going to move into the renal tubule system. The vast majority, over 99% of it, over 99% of what gets filtered out is going to get reabsorbed. So most gets reabsorbed back into our bodies. Urine production rate. Urine production rate is how much urine is produced in a given time, and it has clinical significance. In order to determine the urine production rate, you need to know two things. You need to know how much urine was produced. So in lab, we urinated into our containers that had the graduations on it that showed how much volume was in the container. That's how we figure out volume. And then for time, we think back to when was the most recent time we urinated. We went to the bathroom. And we get time. So right here, 36 milliliters produced over 30 minutes. 36 milliliters divided by 30 minutes gives us 1.2 milliliters per minute. A normal urine production range is 0.5 to 5 milliliters per minute. So it's amount produced divided by time will give us the rate. The combination of filtration, reabsorption, and secretion determines the concentration of various ions in plasma and urine. So remember, it's the combination of what gets filtered, what gets moved into the tubules, what gets reabsorbed back into peritubular capillaries, and what gets secreted from the peritubular capillaries into the renal tubules that eventually determines what gets excreted, what becomes a amount of solute that gets excreted by the kidneys. So pH. So the kidneys help maintain a proper pH within our bodies. pH is determined by hydrogen ion concentrations. Urine pH is lower than blood because of metabolic acids. So CO2 and lactic acid in our blood makes it a little more acidic. And it's more concentrated. Most fluid gets reabsorbed, so it leaves the solutes within the tubule system. Specific gravity. Here's a good question. How did we figure out urine pH in lab? We used a test strip. So it's a piece of paper. You dip the piece of paper into the urine sample, and then it will change colors, and you can compare the color it changes to a set of known colors with known pHs. Specific gravity. So specific gravity is a measurement of well, really it's a measurement of density. But water or urine is water with stuff dissolved in it, and the stuff that's dissolved in it makes it more dense. So the more stuff that's dissolved in your urine, the more dense it's going to be, the higher the specific gravity. The specific gravity of water is 1.0, because it's a measure of concentration. Increased concentration, you increase specific gravity. Increased concentration, increase specific gravity. So typical range, 1.005 to 1.04. What did we use to determine specific gravity in lab? What tool? What was the process? We use the refractometer to figure out specific gravity in lab. All right, so typically, we've all gone to the bathroom before. Typically, when your urine is more clear, you have a lower specific gravity, and the darker the color of your urine, the higher the specific gravity. So know normal ranges, know how we calculated it, know what it is, know that water is 1.0. Clinical examination of urine. So in lab, we looked for the presence of these different things, different things within our urine. We checked for glucose, we checked for protein, we checked for bilirubin, we checked for ketones, we checked for blood or red blood cells and hemoglobin. Know the scientific name for the presence of each of these in blood. All right, so what do we expect to have in our urine? What do we expect to have in our urine? So we do not expect glucose to be in our urine. Glucose gets filtered out. So glucose leaves the bloodstream at the glomerulus and goes into the renal tubule system. But under normal circumstances, 100% of it gets reabsorbed back into our bloodstream. We have a certain number of glucose transporters in our tubule system that can move a certain amount of glucose back into our bloodstream. Diabetics, they sometimes have glucose in their urine. And they have that because there's so much glucose that gets filtered out into the tubule system that their limited number of transporters cannot transport all of that glucose back into their body, back into their peritubular capillaries. So some of it stays in the tubule system and gets urinated out. So it's filtered but completely reabsorbed. Protein in blood, we do not expect to have protein in blood in our urine. Protein in blood, they never leave the glomerulus. They stay within our plasma and they go into the peritubular capillaries and back to the renal vein. Protein and red blood cells under normal circumstances, they go in the afferent arteriole to the glomerulus, into the efferent arteriole to the peritubular capillaries and back to the renal vein. They never enter the tubule system. We expect to see some bilirubin in our urine. That's what makes our pee yellow, in small amounts. And we expect to see some ketones in our urine. The keto diet is very popular nowadays. If you have a low-carb diet, sometimes those levels of ketones will increase. All right, so kidneys filter, reabsorb, and secrete. So renal plasma flow. Renal plasma flow. About 600 milliliters per minute. About 600 milliliters of plasma flows into the glomerulus a minute. A lot of blood flows to your kidneys. A lot of blood flows to your kidneys. 600 milliliters per minute. So 60% of 1 liter, so 1 liter flows to your, 60% of the blood that flows to your kidneys is plasma. About 40% are the formed elements. So 1 liter per minute of blood flows to your kidneys. About 60% of that is plasma. So about 600 milliliters of plasma flows to your kidneys every minute. The glomerular filtration rate, about 20% of what flows to your kidney, about 20% of the plasma that flows to your kidneys gets filtered into the renal tubule system. That's about 125 milliliters per minute. So that's called your glomerular filtration rate. Your glomerular filtration rate is the rate at which fluid leaves your glomerulus and goes into your renal tubule system. Average about 125 per minute. Now, everyone's going to vary a little bit from person to person, but 125 is a good average number. Now remember, a majority of that gets reabsorbed back into our bloodstream, and less than 1% will be excreted. The combination of filtration, reabsorption, and secretion determines the concentration of various ions in plasma and urine. So our kidneys help fine-tune what is in our plasma. Our kidneys help fine-tune the amount of water within our circulatory system. Our kidneys help fine-tune the amount of ions within our plasma. So we need to consider the 3 processes when figuring out what becomes urine: filtration, reabsorption, and secretion. So now I want to get into this idea of clearance, sometimes written as CL. Clearance is the volume of plasma cleared of a given solute in a given time. So how much plasma did we clear of sodium in a minute, for example? How much plasma did we completely remove sodium from in a given time? How much plasma did we completely remove urea from in a given time? The volume of plasma cleared of a given solute in a given time. By cleared, it means you remove that solute from that volume of plasma. Clearance is a non-invasive way to assess renal function. So clearance is determined all the time in clinical settings. Non-invasive, there's no needles, there's no incisions. So clearance is a non-invasive way to assess renal function. Know the definition, know the formula, and we're going to get into how to interpret it shortly. So clearance is the volume of plasma cleared of a given solute in a given time. It's calculated, CL equals clearance, by multiplying the urine concentration by the volume of urine, by the urine production rate. So that's going to give you the amount excreted. Concentration times urine production rate. Going to give you the amount excreted. And you're going to divide that by the concentration in the patient's plasma. So let's look at it for glucose. So these dots are going to represent glucose in the bloodstream. And glucose is going to be filtered out of the glomerulus into the renal corpuscle and the renal tubules. And glucose is valuable. We don't want to urinate out all of the glucose we have in our bodies. So we are going to reabsorb all of the glucose that we can back into our peritubular capillaries. Under normal circumstances, 100% of glucose will be reabsorbed back into our peritubular capillaries. So we have urine concentration. There's no glucose in urine normally. So that value is 0. So we have 0 times urine production rate. We're going to assume it's 1 milliliter per minute. So 0 times 1 divided by plasma concentration, 70 milligrams per deciliter, is going to give us 0. No glucose is urinated out. No plasma is cleared of glucose. So when you see a clearance of 0, that's one way to interpret it. 100% reabsorption would be one way to interpret a 0 clearance value. The other way to interpret a 0 clearance value would be that imagine the situation for protein. Protein never leaves the glomerulus. It never goes into the renal tubule system. It never leaves the renal tubule system, so it doesn't have a chance to become to end up in your urine. So we have a urine concentration of 0 again. So when you see a clearance of 0, it either means 100% reabsorption, so it was filtered and then reabsorbed 100%, or it was never filtered to begin with. I'll repeat that. Clearance of 0 means it was filtered and then 100% reabsorbed, like the case of glucose, or it means that it was never filtered at all and not secreted. So it stayed in the bloodstream the whole time. So glucose is 100% reabsorbed, 0 clearance, 0 clearance. Now let's look at penicillin. So penicillin is filtered out. It's filtered out. It leaves the glomerulus, goes in the renal tubule system. It does not get reabsorbed, 0 reabsorption, and additional penicillin gets secreted out into the renal tubule system. So we have penicillin entering the renal tubules via filtration and via secretion. There's no reabsorption. So we're going to end up with a lot of penicillin in our urine. In fact, let's look at the numbers. So urine concentration, 600 milligrams per deciliter, times urine production rate, 1 milliliter per minute, so 600 times 1, divided by plasma concentration, 4 milligrams per deciliter, is going to give us 150 milliliters per minute. If we had filtration and then no secretion and no reabsorption, clearance is equal to filtration rate. In this situation, penicillin is greater than our filtration rate, which is 100 milliliters per minute in this example. We have we have filtration and secretion at an additional penicillin. When you see a clearance higher than the filtration rate, you've had filtration and secretion of that substance. Penicillin is an example of that. Now, let's look at the situation with urea. So we have blood urea nitrogen, and it is going to be filtered. It's going to leave the glomerulus and go into the renal tubule system. As it goes through the renal tubule system, 50% of it will be reabsorbed. It's not like glucose where 100% gets reabsorbed. It only gets partially reabsorbed back into the peritubular capillaries. So let's look at our math. We have clearance is equal to concentration times volume, so 825 times 1.2, divided by plasma concentration, 12.5, gives us a clearance of 79.2 milliliters per minute. When you see a clearance that is lower than the filtration rate, but greater than 0, it's partially reabsorbed. Some percentage of it will be reabsorbed. All right, so when we want to, let's look at how we can assess this. When we are interpreting clearance, when we are interpreting the clearance value, if the clearance value is less than the glomerular filtration rate, so we're assuming 125 is our glomerular filtration rate in these examples. When our clearance is less than glomerular filtration rate, we're going to have net reabsorption, either partial reabsorption or, if the case is 0, like in glucose, 100% reabsorption. So net reabsorption. Think urea for partial reabsorption. If clearance is greater than glomerular filtration rate, 125 for this example, think penicillin. We have filtration and we have secretion. If clearance is equal to glomerular filtration rate, that means it was filtered and then unaltered throughout the renal tubule system. So it was filtered and then neither secreted or reabsorbed. No net secretion or reabsorption if clearance is equal to glomerular filtration rate. Assumptions. If clearance is greater than 0, the solute was filtered. And if clearance is equal to 0, either, this is what I was talking about earlier, either it was completely reabsorbed, like with the case of glucose, so it was filtered and then completely reabsorbed, like the case of glucose, or it was never filtered at all, like with proteins or red blood cells or white blood cells. All right, here it is again. You can see it kind of written out in a different way. So inulin right here, inulin is a really it's commonly used to assess kidney function in the real world. Inulin is a non-natural molecule used to measure glomerular filtration rate. Inulin is known to not be secreted or reabsorbed, but inulin is filtered, which means that the clearance rate of inulin will equal GFR. If we can figure out the clearance rate of inulin, we can figure out that patient's glomerular filtration rate. And that's what inulin is used for. We're able to calculate the clearance rate, and then we can use the clearance rate to assume or interpret or to guess what the GFR is. So inulin is filtered, not secreted, not absorbed, 100% ends up in the urine. So clearance of inulin is equal to glomerular filtration rate. Inulin, which example does it follow? Inulin follows this right down here. If clearance is equal to glomerular filtration rate, if there's no net secretion or no net reabsorption. So glucose, it's filtered but completely reabsorbed. Clearance is 0. Urea is filtered but partially reabsorbed. Clearance is above 0 but lower than glomerular filtration rate. Inulin is filtered but not reabsorbed and not secreted. Its clearance is equal to glomerular filtration rate. Creatinine is filtered and partially secreted. So clearance is higher than the glomerular filtration rate. All right, everybody, if you have any questions, please let me know. I am here to help, and I will see you in the next video.